I want to thank Cecilia for starting to set up this this really disparity that we see in low resource settings versus high resource settings, like in the US and the UK. The cure rate for pediatric tumors, especially leukemias, is very high, sometimes upwards of 80%. But in low resource settings where they're diagnosed at all, really the outcomes for children with cancer are very poor. For the oncologist out there, sort of this is the normal diagnostic pathway that a tumor may take. You start with your general pathology, going through immunohistochemistry, flow, some cytogenetics, and in some cases, a lot of the good work that's been talked about here is going into molecular genetics and directly identifying fusions and expression profiling. And so our question was, can we use a tool like Nanopore sequencing with a very low capital cost to essentially leapfrog these technologies, right, all the equipment and expertise that's necessary that's not available in low resource settings, and essentially recapitulate as much of that as possible using molecular genetics and sequencing. And so this is our general workflow, right? So with our leukemias, we're getting peripheral blood or bone marrow, right? And we're doing this is full-length cDNA. We've tried a bunch of protocols I'll describe in a bit, but as opposed to a lot of the work we've talked about or that you've heard about, which is DNA-based, we are primarily working in RNA. So we're doing bulk transcriptomics. It's untargeted. And then I'll describe a methodology for taking these bulk transcriptome profiles to subclassify pediatric tumor types. So we have tried all of this, essentially the kitchen sink, direct RNA, direct cDNA, PCR cDNA. I think we did some smarter cDNA at one point across a bunch of devices. And different input material, which I'll talk a little bit when we talk about the difference between leukemias and lymphomas and solid tumors. But all this to say, we've experimented with sort of a huge amount of variation. So again, our motivating principle is to have a technology that will work in really diverse settings, right, with potentially limited training, right, for technologists using this. And so one of the major sources of variation that we see is in the quality of the RNA and the throughput that's achieved on any given day. And so our goal is to develop a platform and Fusion computational tools that is robust to this huge range of variation. So this is to sort of illustrate for you what that looks like. So we've done this in everything from cryopreserved sort of bone marrow, which is sort of our idealized case, all the way to FFPE preserved solid tumor tissue. And you see in the upper left, sort of in these ideal cases, you have this nearly one-to-one correspondence of read length to transcript length, right? These are intact transcripts. And we see all variation of that down to FFPE in the bottom right corner, which are just severely fragmented. So yes, we can do this. We do PCR cDNA primarily on these FFPE samples, and we get reads that are 100, 200 bases. But aside from the length, essentially, we get plenty of them, and those you can anchor to a gene, and we can develop these transcription profiles. You're not going to be able to do direct fusion detection from these really short reads. They're still starting in the poly A. Very often, all we get is part of the 3' UTR. But as long as you can tell what gene it comes from, we can put it into these models. We can use it to build that profile. Again, not restricted to a particular disease or subset. So this is covering essentially most of the spectrum of common pediatric tumors, ex-CNS. So we're not talking about CNS tumors at the moment. Major subtypes of acute leukemia, small round blue cell tumors, and lymphomas. Again, the oncologist will note that some of the lymphomas up here are actually adult. We've done a bit of adult. I'm going to focus on the pediatric results for now. I'm a computer scientist by trade, and so part of the interesting parts was developing this model to do this classification, again, on bulk transcriptomics, and to make this robust to huge amounts of variation. So the punchline later on may be we can do this with as low as about 10 to 20,000 reads. So it is a sparse sampling of the transcriptome, and the methods we use need to be we need to be understanding of that. And so after trying a few things, we landed on an effective method. So this is something called partial least squares discriminant analysis, this PLSDA, which is a variant of PCA that will say rearranges, right, that profile to be maximally discriminative among cases. And so this is sort of an illustration of what this might look like differentiating, say, B cell from T cell leukemia. And we put those, this set of pairwise and sort of one versus all PLS decompositions into a support vector machine. And it produces something, a probability for each type, right? So for B-ALL, T-ALL, and AML in this case. But that's robust to this amount of variation and gives us, in fact, prediction probabilities that are nicely separable. We can do slightly better with some sophisticated, say, convolutional neural networks. And the problem we've experienced with those is that the probabilities do not discriminate. Essentially, they're confidently right or they're confidently wrong. But as we think about down the line, we really want to be able to discriminate, correct answers from incorrect. We'd rather say, I don't know, than give a kid the wrong treatment. So let me orient you to these plots. I think I have a couple more of these. So this is our leukemia classification results into our three major bins, right? AML, B-cell lymphoblastic leukemia, and T-cell. Every dot is one of these cases. So we've done about 300 some odd of these to date. Again, predominantly from cryopreserved bone marrow or peripheral blood. And on the y-axis is their confidence, right? So higher is better. And so see all those dots are correctly classified, and where you see those little X's, these are the ones that we're getting wrong. And so you can see, as we move this toward sort of something realistic in these settings, when you pick and calibrate a confidence cutoff, so around 0.8, you can see how that will help us get rid of some of those these low confidence calls that are often incorrect. The ones that worry us, right, are when this model is confidently incorrect, when it's calling some of these TLL samples BLL. As we've seen these and we go back and forth to pathology, and we say, we keep getting this one wrong, we've gone back, I'm pretty sure we didn't mix it up. And they say, oh yeah, that was this weird case, we weren't totally sure. I'm a computer scientist and I didn't know any better, right? So that sort of thing always happens, but we've been really encouraged so far in how this is laying out. Now remember, these are bulk transcriptional profiles. They're pretty sparse, but all they need to be able to do is tell a B cell from a T cell, and the transcriptional profiles on those are quite distinct. So I'm going to jump now to a more complex problem. Is now we have done several hundred, 500 or 600 solid tumors and lymphomas. Now, as opposed to the leukemias, these are almost entirely FFPE samples. So these are archival samples from UNC and from some of our collaborators at St. Jude. We get these really short reads, but they produce sort of nice profiles that, again, we can tell the difference between, say, neural cells versus rhabdoid cells, right? So essentially what we're seeing is a transcriptional profile that differentiates those cell types of origin. And to do the same thing here, is that on the right side are our these traditional sort of higher prevalent solid tumors, and we're doing nice. They're not as high as the leukemias, but as we add more samples, we consistently see those go up. The classes that we have more information for, this is how machine learning works, we get more and more confidence to build those up. On the left was probably this ill-advised model to try to classify all lymphomas, including many of our adult types, and you'll see that it's messy, but we're doing significantly better than chance at that. I think we had some more recent data. I didn't bother updating my slides, but looking at pediatric lymphoma types alone, there are only 5 or 6 dominant types, and this is doing reasonably well. The question, so these really broad types are enough to move the needle in these low resource settings, right? So that can help them assign the treatment. Again, a lot of the other great work you've heard talk about is getting down into those finer and finer and finer details, including some of these genomic subtypes. Now, like these are treatable, but in these low resource settings, groups we work with in Malawi and Pakistan and India, getting the drugs then to treat more specific types is this next challenge. So it's just sort of this top down. But We have looked to see if these transcriptional profiles, again, can differentiate among common genomic subtypes. You've heard about some of these fusion types. Gross sort of aneuploidy changes are also a diagnostic type for leukemias. And this, again, we're challenged by sort of limited samples in each of these groups, but there is a signal there. So we continue to expand these, start looking a little bit at CNS tumors. Again, they're dominantly methylation-based, and transcripts aren't going to help us very much. But what we really want to do is start seeing how this could actually work in low-resource settings. We have collaborators sequencing samples in Malawi and Pakistan and India, generating that data that varies so much depending on who extracted the RNA that day, and sort of see how it may translate to reality. Thank you so much.